Se ve bien, ¿no? ¿Se escucha? Sí.
Sí, perfecto.
Muy bien.
Buenas tardes a todos y bienvenidos al seminario de la SEBiBC, que hoy va a ser impartido por Francisco Javier Planes. Desde febrero de 2005 hasta agosto de 2008, Francisco Javier desarrolló su tesis doctoral en el Departamento de Matemáticas de la Universidad de Brunel, en Reino Unido. Su tesis doctoral consistió en el uso de algoritmos y modelos matemáticos de optimización típicamente utilizados en el ámbito de la investigación operativa y las ciencias de la computación en un contexto en ese momento bastante innovador y diferente, la bioinformática y la biología computacional. En particular, durante su tesis, Francisco Javier se focalizó en el modelado de rutas metabólicas a escala genómica. Este trabajo de investigación fue, de hecho, galardonado con un premio de doctorado por la Universidad de Brunel. Al finalizar su tesis doctoral, comenzó a trabajar en el grupo de bioinformática de CEIT, perteneciente a la Universidad de Navarra, donde comenzó a desarrollar una línea de investigación independiente en el ámbito de la ingeniería metabólica y la biología de sistemas. En estos 10 años, su grupo ha desarrollado gran experiencia en la integración de distintas fuentes de datos de tecnologías de alta resolución molecular, genómica, transcriptómica, proteómica, metabolómica, todas las químicas, particularmente para extraer propiedades metabólicas, en distintos escenarios de interés en medicina, biotecnología, ecología, toxicología y disciplinas relacionadas. Fruto de todo este trabajo, de todo este intenso trabajo, Francisco Javier ha publicado 43 artículos en revistas especializadas e indexadas, casi todas ellas en revistas del primer cuartil, 41 de ellas, y en la mayoría siendo él o bien primer autor o autor senior corresponding author. Algunas de estas publicaciones en revistas de alto impacto, donde es a veces publicar trabajos metodológicos como pueden ser Genome Biology, Nature Communications o Nature Protocol. En su vertiente más académica y de formación, Francisco Javier ha dirigido 4 tesis doctorales, co-dirigido 3 y actualmente dirige 6. En este periodo, además, ha liderado y participado en 15 proyectos de investigación competitivos, 2 a nivel europeo, 4 a nivel nacional y 9 a nivel regional. Desde marzo de 2017 trabaja con dedicación completa en Tecnun de la Universidad de Navarra dentro del grupo de bioinformática que consta de 15 personas actualmente. En julio de 2019 además fue nombrado subdirector de investigación de Tecnun. Aparte de todo esto, tiene una vertiente también docente, él es catedrático en la Universidad de Navarra y actualmente imparte clases de investigación operativa y biología computacional en el grado de tecnologías industriales y de ingeniería bioquímica. También imparte diferentes asignaturas relacionadas con el Data Science en el máster de Ingeniería Biomédica y el máster de Inteligencia Artificial. Antes de pasar a darle a Francisco Javier la palabra para su presentación, recuerdo a todos los que estáis siguiendo este seminario por YouTube que para poder hacer preguntas al finalizar su presentación tenéis que estar logados en YouTube y podéis hacer las preguntas a través del sistema a través del chat de YouTube. Yo las leeré y se las realizaré a Francisco Javier. A partir del próximo seminario vamos a distribuir el link de Zoom para que podáis vosotros mismos hacer las preguntas, dado que creemos que eso va a ser algo más ágil y más interactivas las presentaciones. Pues sin más dilación, os dejo con Francisco Javier, que va a presentar su trabajo Network-Based Algorithms to Predict Metabolic Vulnerabilities in Cancer. Muchas gracias, Francisco.
Pues muchísimas gracias por la presentación y por la invitación para dar esta charla. La voy a dar en inglés porque hay alguna terminología que me cuesta más traducirla. Y nada, empiezo con la charla Network-Based Algorithms to Predict metabolic vulnerability in cancer. Metabolism is still a hot topic in cancer research. Metabolic reprogramming was in fact defined as one of the hallmarks of cancer, but we still have very papers right now being published in this topic. For example, Rabinovich recently published or validated in vivo the popular Warburg effect, but with a different perspective. So it's still a very important topic in cancer research. So it's an opportunity. Metabolic reprogramming is an opportunity of identifying biomarkers and drug targets for cancer cells. Our approach is slightly different. We start from network-based perspectives. Metabolic networks integrate thousands of reactions from very different databases, and in order to define the prior knowledge of metabolism. Currently, the most updated human metabolic network is in Metabolic Atlas in Human 1, where you can find reaction, metabolites, genes, and proteins involved in human cells. Our conceptual network approach to identify metabolic vulnerabilities is quite simple, simpler than deep learning approaches, for example. Our objective is to block essential metabolites for proliferation. In these reconstructions, in this metabolic reconstruction, we typically have defined some metabolites that are essential for proliferation. So here are
sorry, Francisco, sorry, we don't see the presentation going. It's just the first slide. It's not in now. Now it's fine. For some reason, it was fixed. Now it's fine. Yeah, but we don't see we don't see it in full screen mode. We see it with the maybe it's the way you have shared the screen.
Comparte toda la pantalla. Se relega stop sharing igual.
Stop sharing. Stop sharing y ahora le doy.
¿Cuál es Zoom?
Okay. Y le doy.
Compartir pantalla y toda la ventana.
Vale.
Intenta que se a la derecha. Tendrás que clicar y se selecciona.
Okay. Okay.
Now it's perfect. Thank you.
Okay. Thank you very much. So I was saying that our approach is simple, is to identify ways to block essential metabolites for proliferation, assuming an input metabolites from growth medium, and the gene-protein reaction rules that are defined in the metabolic networks, and available genomics, transcriptomic, and proteomics data. That's our conceptual network approach. For our approach relies on the concept of so in this talk we're gonna see three different sections. First, we're gonna talk about our genetic minimal set approach. Second, I'm gonna present our new tool called genetic minimal set tool. And third, we are going to talk about this approach and the nutrient microenvironment. So genetic minimal cassettes are a group of metabolic genes that, when simultaneously inhibited, render cellular proliferation impossible. In other words, at least one essential metabolite is blocked. Consider the following example in which we have this input of this example metabolic network. We have our target gene, that is metabolic E. Sorry. We have this target metabolic gene, that is metabolic E. We have the gene-protein reaction rules that define the relation between reaction protein and genes. And eventually, we identify the ways to block metabolic E. Here we have three different ways. We can block the synthesis of metabolic E with these three different strategies. One strategy is to inhibit gene 2 and gene 3. Another way is to inhibit gene 2, gene 4, and gene 5. And another strategy is to inhibit gene 2, gene 4, and gene 6. In this example network, it's relatively simple to identify these genetic minimal cut sets. But when we have network with thousands of reactions and thousands of genes, we need sophisticated algorithms. We develop different algorithms to extract efficiently these genetic minimal cut sets using optimization theory, duality theory, and linear algebra. In fact, we implement this algorithm in the Cobra toolbox, and that was published in Nature Protocols, and also all the technical details in bioinformatics in order to clearly define. I'm not going to give mathematical details about these algorithms, but the idea of this algorithm is to extract efficiently the genetic minimal cut sets for complex networks. Somehow, genetic minimal cut sets generalize the concept of synthetic lethality for metabolic networks, but we use higher order synthetic lethal interaction. We can have minimal cut set with 2 genes, with 3 genes, 4 or 5 genes. Somehow it's a network-based synthetic lethality approach. Once we have these genetic minimal cut sets, then we can identify metabolic vulnerabilities. Consider the following, the same example as we were previously discussing, but now we have computed the genetic minimal cut sets and we have transcriptomic data. Some of the genes are lowly expressed and other genes are highly expressed. We can see here that gene 2 is an essential gene because gene 4 and gene 5 are not expressed. Repeating again the idea, a genetic minimal cut set define a subset of genes that if all of them are inactive, then proliferation is not possible anymore. So if we have 3 genes and 2 of them are not expressed, then the other gene, in this case gene 2, is in a specific vulnerability for this condition. In our early paper, we take 30 high-quality Achilles cell lines, the top 20 essential genes and top 20 non-essential genes. This was somehow a gold standard to validate our algorithm. It was using large-scale sequencing or genetic screening data. And we obtained very good results at that time, superior to other algorithms for conducting the same task. In that paper, we demonstrated the essentiality of R-arm-N1 in different multiple myeloma cell lines, given a particular signature. Here we have that this gene is essential according to expression of this other subset of genes. And we tested, we in vitro validated our signature for 4 different cell lines in myeloma. 3 of them were responder to R-arm-N1 inhibition, and the other one was not responder. So this was our first approach using genetic minimal cut sets. Which limitation we found after publishing this paper? First, we need a more automatic application and visualization of this algorithm in a more intuitive and friendly environment, particularly for experimentalists, for researchers in cancer research that were not familiar with our algorithm. Second, we needed a reduction of computational time and to avoid time-consuming optimization algorithm. Third, it was published Human 1, that was the latest metabolic reconstruction of human cells. And there was also challenges to adapt our previous approach for that problem. And finally, our earlier approach was based on microarrays experiments, and we need to go use the RNA-seq data. So with all these challenges, we built a genetic minimal cut set tool that is currently can be accessed, is publicly available for everybody. Is currently this work is currently under review with one of researchers in my group, who is Val Carthel. And in a sense, GMC2 is a web tool application for predicting metabolic vulnerabilities based on human one, RNA-seq data, and genetic minimal cut sets. With this tool, we can predict cancer-specific metabolic essential genes, but also lethal double knockouts. The tool has been developed using the signing package in R, and it's available in Amazon Web Service. So where is the idea of this tool? In a sense, we pre-computed a database of more than 160,000 genetic minimal cut sets from human one. Human one define different basic metabolic tasks, and we adapted our previous algorithm to compute genetic minimal cut sets for the different tasks in human one. So in a sense, we calculate for each task a number of genetic minimal cut sets, and we integrate it all together in a single database that is available in our tool. We can see here that in our database, we have 235 essential genes, but also we have synthetic lethal with pair of genes, 1,555. We have also triplets, and etc. Okay, so we pre-computed a database with thousands of minimal cut sets. When we got this minimal cut set, the next step is to project available omics data. At this moment, we only have RNA-seq data, but we are preparing also the tool to integrate other type of omics data, proteomics, genomic, etc. The idea is with RNA-seq data is transformed into highly and lowly expressed genes. Which genes are active and which genes are inactive? In the literature, there are different methods to conduct these tasks, but we developed our own strategy, exploiting the property of genetic minimal cut set that at least one gene must be expressed to support proliferation. With this idea, assuming that every minimal cut set gives us at least one highly expressed gene, with this we establish a threshold for which genes are active and inactive. Once we have RNA-seq data transformed into high and low expression, then we can now predict metabolic vulnerabilities and identify essential genes and lethal double knockouts. As you can see here, then now is a very simple task. Here you can see the type of output we have in our tool, and also the type of figures we can easily build with our tool. Here we have a genetic minimal cut set with involving 4 genes, and in some of the sample, only one of these genes is expressed. We have also integrated our approach with depth map data, large-scale genetic screens, to validate our predictions. We also, to assess the accuracy of our algorithm, we compare our approach with algorithm proposed by the authors of Human, which is Ponente algorithm, with the same 621 cancer cell line reported in that work. We found that our tool with our threshold is the overall winner. We have a higher Matthew correlation coefficient, as you can see here. We also have a higher accuracy, and we also have a higher sensitivity. So we were very happy with these results, but because we still with this new database, with this new approach, we're obtaining better results than the ones you can obtain with TINIT. And the most striking part is that TINIT took several days to obtain this result, and with our tool, it took us 70 seconds. So our approach is extremely efficient to identify essential genes. We apply our tool to identify metabolic vulnerabilities in myeloma. We got funding from different projects to work in myeloma in collaboration with a group of Professor Felipe Proster. So we have access to a relevant cohort of multiple myeloma patients with hundreds of samples of newly diagnosed multiple myeloma patients. We also work with another cohorts with a limited myeloma samples, but with healthy counterparts from bone marrow plasmatic cells and other cells, another type from beta cell differentiation that we use to evaluate whether the identified targets were selected for myeloma cells. And finally, we also calculate essential genes in different multiple myeloma cell lines. Our objective was to find essential genes in multiple myeloma that are recurrent and specific, selected for multiple myeloma patients samples. We established different thresholds. The most restrictive one was that our target could be non-essential in any of the healthy counterpart in the beta cell subpopulation. And we established different threshold for the COMPASS cohorts and the limited myelomas cohort from our university. And also, we need that at least one cell line that this target is essential in at least one cancer cell line to validate our hypothesis. We could identify 4 essential genes that satisfy this threshold, that were recurrent in myeloma and selected because they were not essential in any of the healthy counterpart subpopulation beta cell differentiation. We focus in one of the simplest targets we could identify that involve CTPS1 and CTPS2. It's a genetic minimal cassette involving only 2 genes, so it was very good for validation purposes. The other 3 were more complex than this one. So we concentrate on this genetic minimal cassette and CTPS1. We found that this, as you can see here, we can find some of the samples. Sorry, some of the sample were in the cohort from our university where CTPS1 is essential, but because CTPS1 is expressed and CTPS2 is not expressed in some of the samples. In the rest of these samples, both genes are expressed. In the COMPASS cohort, we found a high percentage of patients where CTPS2 is not expressed, so CTPS1 would be a selected target. And the same was found with different multiple myeloma cell lines. We validate our results with 3 different cell lines. 2 of them, KMS11 and 8929, were sensitive to CTPS1 validation, but KMS12 is not sensitive because CTPS2 is expressed, as you can see here. We conducted proliferation cultures with selected inhibitor of CTPS1, which validate our prediction with our tool. So What's next? Now we go to the final part. We have talked about our early genetic minimal cut set approach. Now we have built GMSCS tool that is based on human 1 and RNA-seq data to predict essential genes and double knockouts. It's an extremely efficient algorithm because we pre-computed a database of genetic minimal cut set and we validate our approach in multiple myeloma. So the question is, what's next, and how can we improve this approach? We are currently working in 4 different strategies. One is to identify cancer-specific essential metabolites. So far, we've worked with the essential metabolites that are given in human 1, or the basic metabolic task defined in human 1. And we are investigating if we can build a cancer-specific essential metabolic task, or cancer-specific essential metabolic task, essential metabolite. And this is one of the things is working one of my PhD students, Daniel Laverri. Second, we are working in trying to integrate regulatory and signaling pathways with our approach, and we have just submitted an article about this with this work with the PhD of Naroa Barrena. It's under review now. And also, we are now working to how to incorporate single cell data in our approach. But the one I'm going to concentrate is to assess the importance of nutrient microenvironment and the identification of nutrient dependencies with our approach. This is going to be we are working with different strategies to improve our approach, but I'm going to talk about the importance of nutrient microenvironment to identify metabolic targets, and also how can we exploit the microenvironment to identify nutrient dependencies in cancer. Consider the following example. Here we have this specific culture median, where this network with this culture median, and we have 2 different genetic minimal cut sets. And as you can see here, if we block gene 1 and gene 2, then the target metabolite is blocked. If we now change the culture median, we have now an additional escape pathway, an additional alternative pathway that changes the essentiality of this synthetic leaf. In particular, you can see here that with these metabolites, these 2 genes are not synthetic lethal anymore because we have an alternative path. And this is the fact that this is the idea that we exploit in this paper. We extend the concept of genetic minimal cut sets to include not only gene knockout but also nutrients deprivation in the environment. So now with this approach here, we have this genetic minimal cut set, but the previous synthetic lethal, now we have the knockout of these 2 genes, but also the deprivation of these metabolites from the medium. So the computation of, we call it nutrient genetic minimal cut sets, the computation of these strategies is quite direct from our previous algorithm. We only need to add, in addition to gene knockout, also the exchange reaction from that model, the input of nutrients. So with the same algorithm backup, we can compute genetic minimal cut set, but also nutrient genetic minimal cut sets. Following this approach, we conduct this experiment where under RPMI culture medium, the knockout of the hydrofolate reductase, this gene is completely essential. If we knock out this gene, proliferation is not possible anymore. And we tested with 3 different cell lines and with a well-known inhibitor of this protein, and we validated that this target is essential. However, we found with a more general growth medium, we found that we can identify an alternative pathways with adding into the growth medium other nutrients. And we conducted this experiment. We identified the nutrients that could explain the resistance to these metabolic targets, and confirm this computational prediction that if we add in the growth medium thymidine and epoxanthin, then proliferation is rescued. So this is important because when we define metabolic targets, when we try to generalize this strategy to in vivo scenarios, we can find resistance because of a more complex tumor microenvironment. And here you can see a summary scene of why these 2 metabolites generate resistance to the inhibition of the hydrofolate reductase. At the same time, we can our approach can explain resistance. We also concentrate on the other way around. Can we predict nutrient dependencies of cancer cells? And we came across with this work of Javier García Bermúdez, where he reviewed different nutrient dependencies in cancer. One of the most popular ones is the asparagine dependence in different leukemia cells, because one of the essential enzymes for the de novo synthesis of asparagines is lost in some of these leukemia patients. So somehow this is a clinical validation of our nutrient genetic minimal cassette approach, because here if this enzyme AES-NS is lost and we remove asparagine from the growth medium, proliferation is not possible anymore. And in fact, this is used in clinics. We have in this type of leukemia, we have asparagine enzyme given to the patient to remove the asparagine from blood, because this enzyme is blocked. So with this idea, we try to calculate the different nutrient dependencies in cancer cell lines. So we calculated with RPMI growth medium and Recon 3D, which was the previous version of Human 1, of the earlier version of human metabolic reconstruction, and we calculate some nutrient genetic minimum cassettes. And we identify, in our approach, we identified some of them that were known in the literature. One of them is the asparagine, history I've just told you, but also the arginine and glutamine. We could identify, and in fact, in that work, we conducted the experiment using pool RNA studies with, for example, here, this is with high and low asparagine availability, and they identify the same result here, that these are cancer cell lines sorted by the expression of AES-NS. Okay, so cell lines that cannot produce asparagine, the pathway of producing asparagine is not expressed. They are completely dependent on the extracellular arginine. Okay. And they also validate, they conducted the same analysis for arginine and ASS1 and glutamine and gluel, which is, these are 2 enzymes involved in the synthesis of these 2 amino acids. So having validated these three interventions with literature, we focused on these two that were not, we found at that time less information about them. So we conducted the following simulation, where we take we establish a threshold of expression for the genes involved in the cholesterol dependencies. Here we have around 20 cell lines where these three genes are lowly expressed, and another the same number of cell lines where these three are highly expressed. And we use as proxy for cholesterol deprivation the inhibition of LDLR protein, and we take information from that. And we could validate that not only we could validate that in lymphoma and different tumors that is where they have the cholesterol dependency, they were more essential to the inhibition of LDLR. We conducted the same strategy for minocytol. In this case, it was simpler and perhaps easier to understand. The idea is cell lines where isin A1 is not expressed, then it's going to be very dependent on minocytol. So if we remove minocytol from the growth medium, then proliferation is not possible, because we can have data, experimental data with from minocytol deprivation. We use as a proxy the inhibition of one of the transporter of minocytol, and we obtain the same results. We obtain similar results. We identified some specific tumors that were very dependent on minocytol, particularly in acute myeloid leukemia and some another leukemias. In this case, we conducted some experimental validation. Here we have these are with three cancer cell lines. In the red one, the isin A1 is highly expressed, and if we remove inositol from the growth median, nothing happens because you have an alternative pathway for producing minocytol. But in this case, in this cell line, the expression this is TPM, the expression of isin A1 is not expressed at all. So if we remove inositol from the growth median, proliferation is decaying. Okay, it takes time to start decaying, but eventually decays. So these are unpublished results that we are working with additional cell lines and additional experiments. So in conclusion, to end with, our approach, genetic minimal cassette, is an efficient and accurate network-based algorithm to predict metabolic vulnerabilities. We've shown that our approach is more accurate than other methods in the literature, more accurate and more efficient. Second, our tool is freely available and is ready to use, and we will be happy to see other researchers using our tool to identify cancer-specific essential genes and lethal double knockouts. I haven't mentioned our analysis with lethal double knockouts because of lack of time, but it's also an interesting topic, and the large-scale combinatorial genetic screening are proliferating, and we are also studying this type of data. And finally, our nutrient genetic minimal cassette approach can be used to assess resistance to metabolic targets, but also to predict nutrient dependency in cancer. And thank you very much for all my team at Technion. Íñigo Paulaz and Luis Tobalina are not in our group anymore, but Luis Valcárcel is is one of the main authors of this work, and all the PhD students, Aroa, and Carlos, and our collaboration with CIMA and University Hospital, and of course all the Cobra team. We've been working with them for several years, and all the institution for funding our research. Thank you very much.
Okay, so thank you very much, Francisco. As I said before, since we have a little delay, I will start with some questions to see if someone writes something in the chat. So, can you give us some numbers about the computing time for and what is the maximum number of metabolites you can have in a network?
The maximum number of metabolites
that you can model in a network, yeah.
We are working with thousands of metabolites. The human one, I think it's involved around 4,000 metabolites, 5,000. Yeah, it's like human one involves 12,000 reaction and 8,000 metabolites. So we can work with really large metabolic networks, and we can work with network larger than that. Yes.
Okay, but the networks you were showing in your presentation were most of the time like 3-4 nodes. It was just an example from a zoom in from a bigger network, or is this the maximum number?
No. It's just an example network that all the results we show were computed with human one, which is the late metabolic reconstruction of human cells, which involves 12,000 reaction and around 8,000 metabolites.
Okay, great.
The network I showed it was just a way to simplify the explanation of the algorithm.
And for this network that you made with 12,000 reactions, 8,000 nodes, what is the computing time for that?
With our strategy, depending on the task you want to carry out, in genetic minimal cut set tool, our strategy was to compute all the genetic minimal cut set we could, and it took us like 1 month to calculate all the genetic minimal cut sets. But this was a big effort to have all the genetic minimal cut set pre-computed for all the metabolites we have in our study. Once we calculated once, then we only need to project and map experimental data. So we did a huge effort at the beginning to calculate all the minimal cut sets, and then we only need to have to map experimental data. For this reason, now you take sample from different tumors. It takes us like 1 minute to calculate essential genes and 2-3 minutes to calculate little double knockouts, because we pre-computed all the genetic minimal cut set as well. If you want to do something more specific, or you want to concentrate on different metabolites, that in fact we are working in that direction, then you need to calculate genetic minimal cut set for each different metabolite. And this also depends on the length of the genetic minimal cut sets. It could take genetic minimal cut set of size 1, 2, 3, and 4 is relative, not too expensive, but going beyond size 5 it could take several days to compute genetic minimal cut set in this network. But we think that we are confident that with the database we have of genetic minimal cut sets, we can identify relevant metabolic targets. And the idea of our tool is to avoid the calculation of genetic minimal cut sets with optimization algorithms that are really expensive.
Mm-hmm. Thank you very much. Let's see if someone wants to ask something else. They are shy at the moment. I was wondering about the application of transcriptomics data, how you implement the transcriptomics data into these metabolic networks. Have you, because if you try to apply a similar approach to model real actual transcriptomics networks based on, for example, Hill's equation, it should be a similar problem. Maybe you can also use the same approach that you are using for the metabolic networks. I think some groups have used something similar. I don't know if you are aware of that.
Using which type of network? Sorry, regulatory network?
Yeah, exactly. Regulatory network, where the interaction between the nodes should be based on the Hill's equation. That is actually kind of the inverse of the equations you showed for the reactions between metabolites, because this is a monotonously increasing curve, the Hill equation. I don't know if you're familiar with that.
Really, but I think it's a different approach. Our approach is completely structural networks. We are not using mechanism of enzyme kinetics in our approach at the moment.
Okay.
It's more steady state approach assumption, and enzyme kinetics not used or mechanistic equation are not used because they are difficult to measure for a large number of reactions, and we typically don't have the information for all these type of reactions. But I'm aware that the building of all large network with all this information is growing. That's not at the moment for human metabolic reconstructions.
Okay. So I think we don't really have more questions. Let's see if next time logging into Zoom, people are less shy and they can ask themselves. But it was a great seminar, Francisco, and thank you very much for your time and for a great presentation.
Okay, thank you very much.
Thank you.